This is the Chapter 9 Proteins, Part 1 Commentary. So in this commentary, we discuss proteins and how to build very crude small proteins we call polypeptides. So in this slide right here, this gives the overall function of a protein, and as you can see, the functions are diverse. Typical human cells contain 9,000 different proteins, each with its own specific function within the cell, and they can range from a variety of different, for lack of a better word, function. So you have one of the most popular examples where proteins can act as catalysts. Whenever they act as catalysts, the special name for these are enzymes. So here are a variety of enzymes. Each enzyme has its own specific name. Again, these are all common names, and the common names come from the either the function or where they're discovered at, a variety of different reasons. So if you come over here, these are the different purposes. So for example, cytochrome C, it transfers electrons. This is found in the electron transport chain, which we'll research at a later time. DNA polymerase, it's a protein which replicates and repairs the DNA. We'll study that as well. So they cover catalysis, structure, storage, protection. So proteins make up the core of our antibodies and in general our immune system. Regulation. So proteins can serve as hormones, nerve impulse transmissions. So we have receptor proteins for neurotransmitters. We have movement or transport proteins, and they can transport such popular molecules as oxygens, which is we find in hemoglobin and myoglobin. You're responsible for knowing what I'm about to highlight. They're responsible for these numbers. Human body contains 100,000 different proteins, and the protein size can vary from 50 to 27,000 amino acids. They're often classified as what we call colloids. So they're really, really big molecules, so they tend to be too large to pass through cell membranes. So they tend to be made within the cell, and they tend to be made either on the inside or of the cell, and then they exit out, such as through exocytosis or through another means, but they tend to stay within the barriers that they're made. So let's go ahead and go on. In this case, here's an example, what I'm highlighting right here, of a protein. In general, protein and polypeptide is used interchangeably. I do want to point that out. It really depends on the number of amino acids. So if you were to go forward, bit a couple slides. So polypeptides, these are proteins which have less than 50 amino acids. If you have greater than 50 amino acids, then they're proteins. So it just goes along with the number. Less than 50 amino acids, we classify it really as a polypeptide. Greater than 50 amino acids, then we use the term protein. But keep in mind, many biochemists, they use those terms interchangeably. Oftentimes, I do the same thing. So let's go ahead and go back a bit. Here we have our protein. Basically, it looks like a piece of wet noodle, which is wrapped up on itself. If we were to sit there and go in reverse and lay out the strand of molecular noodle, so to speak, we would see it's made up of individual amino acids. So to go in backwards, here's an example of various what we call amino acid residues, which are all linked together via what's called a condensation reaction. And if you want to see what they originally looked like before the condensation reaction, this is what they look like. This right here is a amino acid. An amino acid has some distinct features. It has a amine on one side, and it has a acid on another. Hence, amino acid. Notice it has a central carbon. So we refer to this as the alpha carbon. And on the alpha carbon, we have what's called a R group. This is also known as a side chain group. The side chain group gives that amino acid its unique characteristics. So if you notice on the other amino acid to the left, it has the same exact functional groups. Here, it has the alpha carbon. The only difference, which I'll go ahead and I'll highlight in blue, the only difference is that R group. And again, that R group gives it its unique amino acid characteristics. It also is responsible for why it's named the way it is. So on the right-hand side, because of the R methyl group, we give this the common name alanine. Because of the glycine having that hydrogen, that's why we give it the name glycine. Often what we'll do is we'll go ahead and if we have them combined via what's called a condensation reaction. Why is it a condensation reaction? Because it generates water as a byproduct, hence a condensation reaction. Often when we have it like this, then we'll have a compound name, in this case glycosalanine. My point in this slide, though, in the end, is amino acid type and sequence determine the overall shape of the polypeptide protein. So, in order to have this specific protein, we need to have a proper sequence. And I'm just going to go ahead and label each of these colors. But for example, it might need to be A, B, C, A, B, C, A, B, B. My point in this is just to point out that there is a specific sequence. If I were to sit there and change one of the monomer unit letters, so to speak, and change this one over here, change this one over here to a D, for example, then we would refer to that as a mutation, and this protein would no longer work. So it requires that you have a proper amino acid type and you maintain its proper sequence for this protein to occur. Other key words in here are peptide bonds. So if we take a look at these two starting amino acids, notice how you have a alcohol from the acid and the hydrogen from the amine. They come together forming a water, hence condensation reaction. They've now created a brand new amide, and in biochemistry land, this is known as again, peptide bond. Hence, when we have multiple peptide bonds, then this is where we get the name polypeptides. Let's go ahead and move on. So amino acids, in summary, they contain a amine group and a acid, hence the term amino acid. Notice they have that central carbon again, a alpha carbon. On the alpha carbon, it has that very distinctive side chain. Depending on what side chain you have, it again determines its amino acid characteristics, and it's this is what is responsible for giving it its unique name. The side chain is responsible for intermolecular forces. This is what creates the polypeptide stickiness. So for example, if we were to sit there and go back a slide, here we have our wet noodle. When it wraps up on itself, it actually wraps up due to the intermolecular force. I know it's messy, but intermolecular force, side chain, stickiness, for lack of a better word. We'll investigate that towards the end of the slides. Amino acids we often refer to as Zwitterions. And this is because they have a internal acid-base reaction. Basically, I'm sorry, I shouldn't say that word. In general, here we have a dry amino acid. When you sit there and you put it in water, or make it a aqueous amino acid, the acid portion is going to lose its hydrogen. It's then going to go ahead and transfer it to the amine portion. Consequently, the acid portion is going to have a negative charge. The amine portion is going to have a positive charge. So this is the general state of a amino acid when it's found in neutral water or in the aqueous water of a living organism. So, isoelectric point is a pH at which an amino acid has a net charge of 0. So that means the positives balance out the negative charges. So in this case, inside a living cell, pH is very important. We need to maintain a specific pH so that way all of our positive and negative charges form properly and balance each other out. So keeping it neutral. So if you screw with the cell or a blood's pH, then you have various unwanted results on proteins, because then you start affecting the amino acid charges. In general, this is a very overall hard concept. Go ahead and review this definition right here. Short of that, I'm not going to go into too much detail. But bottom line is, we need to make sure that we are at the isoelectric point for these biochemical related amino acids, so that way the positive and negative charges balance out properly, maintaining the stability of the polypeptide. Here is another summary of the formation of a polypeptide. Again, if it's less than 50 amino acids, a polypeptide. If it's greater, then it's a protein. So polymerization, aka condensation reaction, because it gives off water. So two names for the same thing. It generates a brand new amide. The biochemical name for this is a peptide bond. A peptide bond. Multiple peptide bonds result in a polypeptide formation. So here it's highlighted here. What I did was I tried to highlight that in my big picture of a single strand of polypeptide. Notice again, and I'll circle this in with a blue pen, these are side chains. These side chains result in the intermolecular force stickiness of the polypeptide, resulting in the polypeptide wrapping up on itself like a wet noodle. Keep in mind, not only does it need the proper amino acid type and proper sequence, but those side chains are responsible for making sure it wraps up in a specific conformation. If it doesn't wrap up in a specific conformation, well, it needs to wrap up in a specific conformation in order to function properly. Oops. So let's go ahead and move on. Repeating myself, go ahead and scratch this out. That's a typo. I didn't have a chance to change it, so we're going to change. So ignore that. Here we have a peptide linkage. Again, three different names for the same thing: peptide linkage, peptide bond formation, and this was all due to a condensation reaction because it gave off water. I'm beating myself to death. I do want to point out here we have our glycine. Here we have our amine. Even though it's in its ionized form, it's we're going to have 2 hydrogen from this amine. We're going to have one oxygen, and they're going to form a water molecule, and then it's going to go ahead and form a amide or peptide bond. This occurs whether it's aqueous, well, this occurs in its aqueous form, obviously. Please do not get wrapped up on how the atoms come off. It can be a little bit tricky, and then if you want to, I can show you a separate animation in class. But focus on how it forms the final polypeptide. You'll be responsible ultimately for going from the before and before to after on this. Let's move on. So we talked about polypeptides, proteins, amino acid residues. This is when the amino acid is now part of a polypeptide or protein. So in this case, when it's actually formed a new polypeptide, we can think of these individual amino acids as amino acid residues. Now, we have a north side and a south side, so to speak. We have a starting side and an end side. The starting side is a N-terminal residue. Why is it N-terminal? Because at the beginning of it, you should have a positively charged amine at the beginning. And at the end of the residue, you should have a negatively charged carboxylic acid ion, aka carboxylate. So that's the N-terminus, that's the C-terminus. For all polypeptides and proteins, you always have the N-terminus and C-terminus, well, at least for this class. Here we have examples of 4 types of amino acids, and I specifically chose these amino acids because they represent one amino acid from each of the classes, from each of the main amino acid classes. As mentioned earlier, what gives it its unique characteristics, its unique name, and ultimately why it's classified in that class is due to the side chain. Notice in what I just circled in blue for leucine, if you notice, this side chain is nothing but hydrocarbons. Hydrocarbons, if you reason it out, are London forces. They're overall nonpolar. Consequently, it's a nonpolar amino acid. If we go to aspartic acid, notice at the very end we have a carboxylic acid. That's where we get part of the name, aspartic acid. Consequently, we call these acidic amino acids. Now you should be able to figure out that the bottom one is going to be a basic amino acid, and then the one on the left is going to be a polar amino acid. Why is it polar? Because hydrogen bonding, therefore it's polar. On this one right here, realistically, when it's inside water, it should look like this, NH3 with a positive charge, So here's another one I didn't have a chance to change, but it really should be a positive charge when it's in aqueous. This one over here, this one should be a negatively charged when it's in aqueous. So my point on this is assume that these are neutral, dry amino acids. And if we were to sit there and stick them into water, then we would have to lose the hydrogen on this particular amino acid and then gain a hydrogen on this other amino acid. But assume that all of these are neutral amino acids in this slide. On the left-hand side, it doesn't make a difference whether it's neutral or not. They're not going to gain or lose any hydrogens. They maintain their overall shape. So when it says the word non-Zwitterion form, that's actually code for saying these are all dry amino acids. And when they're dry, the ones on the right-hand side are neutral to begin with. The other thing is, and I want to bring this up, if you notice for each of the amino acids, these are also neutral because they're dry. So many little characteristics, but so that's another reason why it's non-Zwitterion, because positive and negatives, notice there are none. Consequently, they don't balance each other out either. So this is neutral. Consequently, these are all example of, again, dry amino acids. And I know somebody's going to ask a question on this, so I should bring this up immediately. What happens, for example, if this one right here were aqueous? Then it would be written like this right here. I'll go ahead and I'll pop this up really quick. It'd be written like this. I know it's a little bit messy. So here we have CH2, CH, CH3, CH3. Oops, CH3. So notice, even though there's no changes to the side chain, when it's aqueous form, it's put into water, you have your positive amine, aka ammonium, and you have your negative charge, because it lost a hydrogen, carboxylic acid, aka carboxylate ion. So you would have to be responsible for drawing this in its aqueous form. So please be careful. I could say the word dry or I could say the word aqueous when I ask you to sketch these four. And now that I'm thinking about it, if I wanted to sit there and draw this R chain, another way I could sit there and draw this entire molecule, and I'm just going to go ahead and draw it in its aqueous form. Let's go ahead and draw this one just to get it out of the way. Here we have a NH3+. Here we have a carboxylic ion, negative charged. Now watch what I do here. This is how I get to be lazy, and this is how I keep track of the number of carbons. So 1, 2, 3, 4. So 1, 2, 3, 4. And then here we have on the bottom our ammonium because it picks up a hydrogen positive charge. So that's another way of writing it. And note this is in its aqueous form. So this is in its Zwitterion form. Let's move on. I beat this to death already, but polypeptides fold maintained due to amino acid stickiness. And that's from the side chain intermolecular forces.